Hi, hello everyone, and welcome to the 11th presentation in this webinar series. My name is Andres Sibbles, and I will have the pleasure of serving as your moderator for today's webinar. So on behalf of the USDA Climate Hubs and the Long-Term Agroecosystem Research Network, we'd like to welcome you to this presentation. As you recall, if you've attended other presentations, is that the goal of this series is to provide an update on the knowns and unknowns about the influence of management on soil carbon dynamics in grazing lands of the continental United States. Recall that today's presentation will be followed by a short question and answer session. And so please type your questions for the speaker using the Q&A tool. So that's not the chat tool, but the Q&A tool on your screen. And today's webinar will conclude promptly at 45 minutes past the hour. The title of today's presentation is Depth Distribution of Soil Organic Carbon and Nitrogen Fractions in a Tallgrass Prairie in Oklahoma. And today's speaker is Dr. Alan Franzlubbers, who is a research ecology with the USDA Agricultural Research Service on the campus of North Carolina State University in Raleigh, North Carolina. Dr. Franzlubbers has conducted extensive research in soil ecology and management and has already presented in this webinar series results from his research in North Carolina and Kentucky. And today will talk to us about a study in the Southern Plains in Oklahoma. He has a leadership role in the USDA Southeast Region Climate Hub and serves on the board of directors of the American Forage and Grassland Council. So without more to do, let's listen to Dr. Franzlubbers' presentation.
Good day. My name's Alan Franzlubbers. I'm with the USDA Agricultural Research Service in Raleigh, North Carolina. I'm happy to be with you and share a study about the depth distribution of soil organic carbon and nitrogen fractions in a tallgrass prairie in Oklahoma. This study was conducted in the Southern Great Plains in El Reno, Oklahoma, as part of the Climate Hub network. The United States has a diversity of climatic conditions, and El Reno, Oklahoma is at the crossroads among many of these climatic conditions, so it receives a great deal of variability in its conditions from year to year. And we have the warm, humid region in the southeastern U.S., the cool, humid region in the northeastern U.S., generally the cool, dry region in the northwest, and the arid region, the warm, dry region in the southwest. El Reno, Oklahoma has a typical pattern of relatively dry summers and wet winters. Precipitation is generally greatest in the spring and the fall, shown here in the blue line, and then evapotranspiration occurs dominantly in the summertime with the deficit that occurs in that summertime period. So this affects the growth pattern of native warm season grasses. Yearly precipitation is approximately 32 inches. Average temperature is 60 degrees Fahrenheit, and there's a net deficit over the whole, between precipitation and evapotranspiration over the whole year. This study was conducted because there's a mass conversion of grasslands to cropland, and there was neglect of remaining lands reduced their prominence in the 20th century. Since the turn of the millennium, greater recognition of the grassland's role in conserving soil, promoting biodiversity, stabilizing farming communities, and providing numerous ecosystem services has led to renewed interest in grassland functions. Rotational stocking has been promoted to allow forage stands to rest and accumulate biomass prior to the next grazing event. We wanted to test this out. So the objective was to contribute to the scientific discourse by documenting ecosystem relevant effects of stocking method on soil carbon and nitrogen fractions in a temperate, mostly native grassland ecosystem relic in the central Oklahoma of the U.S. Great Plains region. Our hypothesis was that rotational stocking would lead to improvements in soil organic carbon and nitrogen fractions relative to continuous stocking in response to greater residual forage mass, surface residue cover, and deeper and more vigorous ridding. El Reno, Oklahoma can be characterized as a location in the tallgrass prairie ecosystem of the Great Plains. This study was 850 acres of grazing land and established in 2009. The experimental setup was for 2 treatments, continuous and rotational stocking, 2 replicates of each of these treatments, 130 acres of continuous grazing land, and 225 acres of rotational units. Rotational stocking was with 10 sub-paddocks each, so roughly 20 to 25 acre sub-paddocks, and a stocking rate of approximately 0.6 head per acre, or 1.7 acres per cow-calf pair. Continuous stocking was from March to October. And rotational stocking was with 1 to 3 week-long grazing events per year. The soils were generally austals, meaning that they were mollisols that were of dry nature, but obviously it was in the subhumid to semi-arid climate and variable from year to year. They were freely drained, and a variety of silt loam soils were present, anywhere from Norge to Pond Creek, Kirkland, Pawhuska, Bethany, and most of them were rather thick mollisols, indicated by the PACIC descriptor, and they had relatively active clay fraction in the soil. Soils were sampled in 2009, which was the initiation of the study, in 2012 at the end of 3 years, and in 2017 at the end of 8 years. Our soil analyses included the analysis of depths of 0 to 6, 6 to 12, 12 to 20, and 20 to 30 centimeters at 0 and 3 years. And then these soil sampling depth changed to 0 to 15 centimeters and 15 to 30 centimeters at 8 years. So that was a little bit of difference, and that complicated the analysis somewhat. Soil was dried and sieved through 4.75 millimeters to get a homogeneous sample. Total organic carbon and nitrogen was determined with dry combustion on a ball milled sample. Potential carbon and nitrogen mineralization was determined at 50% water-filled pore space and 25 degrees Celsius. And inorganic nitrogen was extracted in a KCl solution and analyzed for ammonium and nitrate. The results of the study are shown here. Generally, there was a large change that occurred with soil organic carbon high near the surface and declining with depth. And this occurred whether it was in continuous stocking or rotational stocking, and basically this was the initiation. We didn't expect any difference here. We didn't see any statistical difference in our analyses at any of the depths, but we see that there was greater organic carbon near the surface and it declined with depth. This is a natural characteristic of many of upland soils. Soil organic carbon, so there essentially there was no difference between these studies, these treatments. Total soil nitrogen followed a similar pattern. Even at 3 years, there was no significant difference that occurred. And so there was no significant change that occurred over time. Now, if you look at the soil organic matter depth distribution from other studies, we see that there's this similar characteristic of high concentration near the surface and declining with depth. Typically, we'd like to sample a little bit deeper than the 30 centimeter depth or 1 foot depth, and we want to go a little bit deeper to characterize what is the pathogenic contribution versus the management-induced contribution. And we can get that with the root zone enrichment calculation, because at 30 centimeters or at 1 foot depth, we can assume that the baseline condition is relatively stable, and that there's really no influence of management below this level of soil sampling depth. And that any increase that occurs in the top 12 inches is due to management, and that would be what we'd be calling the root zone enrichment. And we sum the different depths within that 30 centimeter depth. And if we look at the root zone enrichment of total soil nitrogen in pounds per acre to the depth of 12 inches in this case, and we see this by years of management, we saw no statistical difference, but we still saw that the continuous stocking tended to increase, and the rotational stocking increased as well. And on average, there was an accumulation of 31 to 74 pounds of nitrogen per acre per year under this management system, under this tallgrass prairie. And it was not really affected by the stocking method, but this shows the range of values that were there. It's complicated a little bit by the fact that we only sampled 0 to 15 and 15 to 30 centimeters at 8 years, and then different depth increments at 0 and 3 years. But these are our best estimates of this change in time over time. If we look at the total soil nitrogen, it was highly related to total organic carbon. And that is typical of most studies. We see that the carbon and nitrogen are typically very closely associated. And from this equation here with the model would suggest that there was a C/N ratio near 16 at the surface and around 10 at lower depths. And that's indicated by a little bit of, if we change this linear regression to a polynomial, we can see that there are points where there would actually be more of a curvature there, and that's why the C/N ratio was a little bit different. But basically, these results suggest that there were no carbonates present in the soil, and that the total carbon was actually equal to organic carbon. Looking at the particulate organic carbon, this is the sand-sized material that's in soil. We see that there was also a strong association between particulate organic carbon and total organic carbon. And there was somewhat of a nonlinear association here, and that was indicated that near the surface, the proportion of total organic carbon as particulate organic carbon was about 40% near the surface, and that declined with depth to approximately 10% at lower depths. So particulate organic carbon can be a significant fraction. Of course, this is material that is coming from the senescent plant material and dung from the animal grazing, and that contributes to the feeding of the soil microbial community that then decomposes organic matter and leaves behind residual organic matter as total organic carbon. We look at the root zone enrichment of soil test biological activity, and we saw also an increase that occurred over time. It was not necessarily a static linear increase, because between 0 and 3 years there was very little change, and then there was some change again at 8 years. But this might be a little bit of a difference due to the difference in sampling depths as well. But in general, we could say that there's an accumulation of 46 to 60 pounds of root zone of this root zone enrichment of soil test biological activity per year. And this would be a relatively small amount of enrichment, but it's something that is enriching the soil due to the grazing management that is occurring. And we'd like to see if this is actually stable over time, or if it continues to increase over time. Looking at the net nitrogen mineralization, we see that there was a strong association between net nitrogen mineralization and soil test biological activity. This suggests that there's a good association, and that there's a predictability of net nitrogen mineralization, which is a relatively complicated procedure to go through. But this is through aerobic mineralization that occurs, and we can see that soil test biological activity is actually well associated, and that there can be a prediction that occurs. And there's strong evidence that this occurs because of nitrogen mineralization is a biological process, and so biological activity by this test would indicate that. And we can see here that these results, the slope of this relationship was had a slope of 0.18, and this coefficient was a little bit smaller than the coefficient that we typically get from eastern United States soils. And that's maybe due to the type of clay that's present in the soil, and that there's maybe a little bit more immobilization that is occurring. Looking at apparent nitrification along with this net nitrogen mineralization, this is probably one of the most peculiar and interesting points of this study, was that net nitrogen mineralization declined with depth, and that is a natural condition that near the surface organic matter is enriched, and then it is less enriched with depth, and that's why nitrogen mineralization declines with depth. But we also saw that typically in cropland soils, we get near 100% nitrification that occurs. But in this grassland soil, we saw that under whether it was continuous or rotational stocking, that the native plant material that's there was leading to some sort of inhibition of nitrification that occurred. And this nitrification inhibition was stronger deeper in the depth, with depth than it was near the surface. So despite high capacity for nitrogen mineralization, apparent nitrification was limited, thereby effectively limiting loss of nitrate from soil, either through leaching or denitrification. And that inhibition of nitrification can be considered a good thing, because this is avoiding these nitrogen losses that would typically occur. And so this might be a characteristic of native grasslands that needs to be explored in other systems too. And understanding why nitrification is inhibited and the molecules that might be present could lead us to different types of land management that we can actually try to inhibit nitrification naturally rather than through chemical additions. So in summary, there was strong depth stratification of soil organic carbon and nitrogen, and this is typical of other studies as well. There were no statistical differences in soil organic carbon and nitrogen fractions over 8 years of differential stocking and management. This was contrary to our hypothesis. We did think that the rotational stocking would lead to an increase in soil organic carbon and nitrogen fractions, but it's something that we tested, and we did not find any difference between these stocking methods. So rather than declare that there will be a change in soil organic carbon and nitrogen, we'll need to measure some differences between these stocking methods in the future as well. There was high initial concentrations of soil organic carbon and nitrogen fractions, and that might be the reasons why we didn't see any difference between their stocking methods, is that the condition of the soil was actually in a relatively good health, and so that there was maybe no apparent or immediate change that could occur. Soil test biological activity and total soil nitrogen were trending greater over time, but additional evaluations are certainly warranted because we did have this change in sampling depth that confounded the results, and so we want to make sure that there's actually a significant change that's occurring over time. And inhibition of nitrification may be a mechanism for nitrogen conservation. And this needs to be explored a little bit further to understand what are how this is occurring in the soil, and if there are certain species in the plant mixture that might be contributing to this inhibition. And certainly the inhibition is not necessarily a negative. It could be considered a positive because it would limit nitrogen losses either through volatilization or denitrification or leaching losses. Well, I thank you for your attention to listen through this study of depth distribution of soil organic carbon and nitrogen fractions in a tallgrass prairie in Oklahoma. I thank my collaborators, which are retired people of Patrick Starts and Gene Steiner, and I appreciate the opportunity to collaborate on the project and analyze the samples, although I was not physically present at the study. And I hope that the study continues under the leadership of Stacy Gunner, the Center Director at the Grazing Lands Research Laboratory. Thank you, and I look forward to your questions and answering them after the seminar. Thank you. Bye.
Andreas, you're on mute.
Thank you, Susan. All right. I was just saying that, thank you, Alan, for that presentation. And Dr. Franzlubbers is joining us from the field today to answer questions. So we appreciate you doing that, Alan, this morning. And we have a few questions. And the first is regarding differences in the plant community composition or species abundances between stocking treatments after 3 to 8 years, if you're aware of differences there.
Well, thanks for the question, and thanks for the opportunity to field them. I am not really aware of the differences in the botanical composition. Unfortunately, as mentioned, I was not present at the study, so I don't really have a good sense of what the results were for the botanical composition. But I believe there was another paper, and if there is anybody from the El Reno lab, Andres or anyone else that could answer that, I'd be happy for them to jump in.
Thank you, Alan. I wonder if Anne-Marie or Brian are in attendance. Please, I think Susan can unmute you and you can help address, provide a bit more details on this question. Susan, do you know if Anne-Marie or Brian are with us?
They are not on right now.
They're not on right now. Okay, very well. So we will provide answers to these questions online. Online on the website. Sorry, eventually. So yep. We apologize about that. Alan, what would you expect to see in low organic matter soils, since your study was conducted in a relatively high organic matter soil?
Right. Yeah, I think that's a good question, and I think the setting, the environmental setting, is an important attribute that we need to characterize for all studies, and for all farms that we are actually trying to relate to. So the initial soil organic matter conditions could be a very important part. When we have, and the issue, it becomes, what are the thresholds when a soil organic matter is high enough where we wouldn't necessarily see rapid change, or a soil organic matter is low and we would see, well, making a significant stocking method change would lead to greater organic matter. I think that's the issue that we face, and maybe the assumption in the popular press is that our soils are so degraded that, of course, changing to a rotational stocking method will only lead to greater organic matter. And that, if we know that those conditions are in, the soil is in poor condition, then maybe that's a good assumption. But I don't think we can make that assumption in all cases. That's why we need to have some assessment of the soil health condition up front, so that we would know what are the conditions. And I think that's where getting more data from around the country on what are the conditions on private lands, on research stations, and characterizing that. So that's going to be useful. So I would say that in a low organic matter soil, we could expect that rotational stocking could lead to greater organic matter.
Thank you, Alan. We have another question here that asks whether, based on your results and summary, do you feel confident in saying it is best to focus our resources, time and money in future testing to the top 1 foot of soil as it relates to soil carbon and nitrogen testing and expected changes based on management?
Thanks for the question. I think that the question has more than just the question that is there. So I think that there's currently our soil testing is typically the top 4 or 6 inches. So in a pasture system. So going to 1 foot is extending that traditional sampling depth, and I think that that could be useful in some cases, but again, I would say that targeting our resources effectively is an important part of the question. When we want to see changes in organic matter, I think the top 4 to 6 inches is probably where we should start. If our resources are really limited, yes, let's start there. If our resources are a little bit more extensive, then getting different depth increments could be useful, because the only way to actually calculate this root zone enrichment calculation is through multiple depths in the profile. You have to have a minimum of 3. And so if resources are available, this could be a very useful approach to actually understand what are the changes due to management versus those from soil formation factors that are inherent in the soil. So I would say that focusing, if the resources are limited, focusing on the very surface is probably an appropriate approach, but that doesn't lead to comprehensive understanding. But if the resources are limited, again, focusing on the surface is okay.
Thank you. This question asks whether from your study, can we infer that there was no change in root growth or soil carbon contributions below 30 centimeters?
Yeah, the below 30 centimeters, there are changes that do occur. We can have chemical changes in the soil properties. We can have roots that penetrate below 30 centimeters. That's a given. The issue is that the steady state level of organic matter is likely not being changed, though. There are roots that are penetrating, and they're depositing carbon and nitrogen substrates throughout this lower depth, but at a very low level compared to the top 12 inches or the top 30 centimeters. That's the issue, is that the extent of rooting below 30 centimeters is simply very limited. The available evidence would suggest that there's at least 75 to 90% of the roots of almost all plants, even of native warm season grasses, in the top 12 inches. So we should, when we realize that, then yes, there are some minor changes that could be occurring below 30 centimeters, but that's where we're using our resources to try to find something that is really difficult to find. So chemical changes do occur below 30 centimeters. Organic matter changes are likely not occurring below 30 centimeters. That's a perspective that I'd like to share fairly strongly, that it's worth the effort to sample below 30 centimeters in some cases, but not as a routine.
Thank you. Alan, another question here. Do you think the finding of lower nitrification with depth could be related to lower nitrogen or difference in soil organic carbon over the same depth?
Yeah, that's a very good question. I'm a little bit puzzled yet about the nitrification issue. So I believe that the evidence is that the biological activity certainly declines with depth as well. The total resources available with depth are also declining. Although this is a prairie soil that has relatively high organic carbon and nitrogen even at 30 centimeters. It's certainly lower than at the surface, but it's still relatively high compared to many other soils. And our soils that do have, typically would have higher nitrification, oftentimes have far less organic carbon and nitrogen in them. So I'm not sure that the total nitrogen is the issue. I believe that it's probably more likely, and of course it's only a hypothesis, that there are substrates in the rooting of these species that are, and it may be very species specific of the botanical composition of what is contributing to this nitrification inhibition, because it seems, at least from the evidence that I've seen, there are other grasses and sometimes woody plants that have this inhibition that's occurring, and we see that also many times in woodlands, but also in warm season grasses. So there must be something going on, because in cool season grasses, we don't see this evidence. At least I don't see it in the evidence of the data that I've been able to measure in our studies. So it seems to be something that's warm season related, but not necessarily all warm season grasses. There must be some biochemical compound that is contributing to this inhibition. That's a hypothesis that needs to be tested.
Thank you, Alan. We have a question about amendments, and the question goes, compost and biochar amendments are being tested by Quivira Coalition on dry lands. Perhaps extending study finding, multi-year carbon sequestration, and so on. So any comments on these amendments on compost and biochar on drylands or pasture land? Yeah.
Yeah, it's a good question. I think amending soil with organic substrates is always a good thing. That's what we intend to do with our pastures, because we want a robust pasture growth. So those pasture grasses and those forbs are sequestering carbon from the atmosphere, and we want them to contribute to organic matter in the soil. So I think organic amendments with biochar and composting, they're going to have value, certainly. But it's an input. It's not derived from that land area. It has to be derived from another location. So that, there's a carbon balance that needs to be calculated of where those substrates came from. But I think in general, compost and biochar in a mix, I haven't done much research myself, but I like the idea. I think that's where stabilization of organic material can really occur, when you have biochar and compost together. So I look forward to hearing more about that.
Thank you, Alan. Is it another question here? Is it safe to assume a more fibrous rooted plant is more advantageous than a plant with a long, deep taproot in regard to carbon sequestration?
It's a great question. I would just, I could almost defer to and say that I'm not a plant ecologist. So, but I think that it's the combination that in native grasslands that are what's important. If you didn't have the deep rooted roots, then it might be limiting what can occur as far as plant productivity. So I think it's the combination that's really effective, of exploring the soil for extracting nutrients as well as absorbing water. So I think that's the key, is that the system working together is more holistic when it has that diversity of rooting attributes. So I wouldn't say that it's one or the other that's more important, but certainly fibrous root systems means that there are more surface areas able for carbon to leak from the roots, either leaking or sloughing off to contribute to the soil. So, it would seem that more fibrous could be more effective, but I really think that the combination is probably appropriate.
Thank you. That concludes the questions. We realize there are a couple of questions on prior use and stocking rates, and those we'll try to get answers to those questions on the website with help from some of the scientists at the Grazing Lands Research Lab. So thank you very much, Alan, for the presentation and for your response to the questions. Please remember, you've seen Susan's messages in the chat. There are some, there's a poll that Susan will put up in a bit, and we ask that if you have time, you answer those questions before leaving. We want to remind you again that this is the last in our 11th webinar series, and please be on the lookout. We are planning to produce fact sheets with each of these presentations, and also a series of short synthesis articles as well. And so as that process progresses and as things become available, we will send out a heads up to all the folks that registered and attended these webinars. So thank you again. We appreciate your comments, and we appreciate you attending this series, and we hope to follow up soon. So happy holidays to everyone.